In this video tutorial, I am going to discuss about regulation of enzyme activity by the change in enzyme concentration. And remember, change in the enzyme concentration can be done by three different mechanisms. The first one is enzyme induction, second is the enzyme repression, and third is the enzyme degradation. Right? Let's try to understand this. See, the concentration of enzyme is dependent on two factors. One is synthesis and second one is the degradation. If synthesis of enzyme occur, then the enzyme is added into this concentration of enzyme, and if degradation occur, then enzyme is removed from this concentration of enzyme. So if there is an increase in the synthesis, it leads to increased concentration of enzyme, and if there is a decrease in the synthesis of enzyme, it leads to decrease in the concentration of enzyme. So increased synthesis of enzyme is called as an enzyme induction, whereas decrease in the synthesis of enzyme is called as an enzyme repression. And similarly, this degradation can also be regulated, right? So let's look at all these mechanisms one by one. So the first point to discuss is the enzyme induction. So as I told you, enzyme induction is nothing but the increased rate of synthesis of enzyme. Now this increased rate of synthesis of enzyme is carried out by some special molecules. These special molecules are known as inducers. Mostly these inducers are either hormones like insulin, glucagon, or they are the substrate of that enzyme, right? Now these enzymes which can undergo this change in the rate of synthesis, they are called as a inducible enzyme. Inducible enzyme. It is also called as a adaptive enzyme. Here one thing to note down is that not all enzymes of our body are inducible or adaptive enzyme. Rather, there are some enzymes in our body which doesn't undergo increased rate of synthesis. Rather, their synthesis rate is quite constant. And such enzymes are called as a constitutive enzymes. So what is the meaning of constitutive enzyme? Those enzymes whose rate of synthesis is constant in our body. Whereas what are the inducible or adaptive enzymes? Those enzymes whose rate of synthesis increases in response to inducers. Right? Now let's look at the examples of enzyme induction. So we have two very good examples. The one is glucokinase, and this enzyme is of the pathway glycolysis. Glycolysis is the pathway by which glucose is broken down. And the second enzyme is the pyruvate carboxylase. Pyruvate carboxylase is of the important enzyme of the pathway gluconeogenesis. Gluconeogenesis is the pathway by which new glucose molecule is synthesized from the non-carbohydrate sources. So here we have two opposite pathways. One is glycolysis, that is breakdown of glucose, and other one is the gluconeogenesis, which is the synthesis of glucose. Now the glucokinase, for its substrate, that is glucose, and one hormone, that is insulin, act as a inducer. That means glucose and insulin both increases the rate of synthesis of glucokinase, whereas glucagon, it induces this pyruvate carboxylase. Okay, so that means glucagon induces the gluconeogenesis pathway. Now let us look at the second regulatory mechanism, that is enzyme repression. The opposite of enzyme induction is the enzyme repression. What does it mean? That decrease rate of synthesis of enzyme is called as an enzyme repressor, and this is carried out by some special molecules, and these special molecules are called as a repressor. So repressors are the molecule which bring about decreased rate of synthesis of enzyme. Here also we have two examples of the opposite pathway. The first one is the pyruvate kinase, which is the enzyme of glycolysis, and second one is the pyruvate carboxylase, which is of the pathway gluconeogenesis. Now this pyruvate kinase is repressed by the hormone glucagon. So glucagon decreases the rate of synthesis of pyruvate kinase, whereas insulin acts as a repressor for the pyruvate carboxylase. That means insulin inhibits or decreases the rate of synthesis of pyruvate carboxylase. Now if you are an astute viewer, you must have noticed that enzyme induction and enzyme repression both are carrying out one very good or very beautiful balance of the metabolic pathways. See, if you compare gluconeogenesis, which is synthesis of new glucose, and glycolysis, which is the breakdown of glucose, right? Both are very well controlled by this enzyme induction and enzyme repression, right? So let's first look at the glucagon. See, if you examine glucagon, you can see that glucagon is acting as an enzyme inducer for the pyruvate carboxylase and in turn gluconeogenesis. So I can say that glucagon stimulates gluconeogenesis, whereas in this case enzyme repression, glucagon is inhibiting this pyruvate kinase and in turn inhibiting glycolysis, right? So glucagon inhibits glycolysis. So glucagon is making sure that when new glucose is being synthesized, glucose should not undergo breakdown. And opposite thing occurs in presence of insulin. See, what is insulin is doing to the gluconeogenesis? Over here, insulin is inhibiting this gluconeogenesis, right? So, insulin inhibits new glucose molecule synthesis, whereas on this enzyme induction, this insulin is stimulating glycolysis, right? So, insulin stimulates glycolysis. So, the presence of glucagon and insulin determines that whether glucose will be broken down or new glucose will be synthesized. So, this is such a beautiful balance which indicates metabolic coordination at its finest point. Now, let us talk about the third mechanism, is the regulation of enzyme degradation. Enzyme degradation. See, we know that all enzymes are proteins, and we had already discussed that how proteins are getting degraded. If you don't know how proteins are degraded, you can visit my previous video and the link is given in the I button. Now in case of enzyme degradation, certain molecules or substances are there which can stabilize the enzyme. So most of the time enzyme stabilizers are the substrate of particular enzyme, then coenzymes or metal ions. Such type of molecules generally stabilizes enzyme, and so they can decrease the degradation of enzyme. Let's look at the example. As an example, we have enzyme tryptophan pyrrolase. This tryptophan pyrrolase, just like any other protein, after its half-life is over, it undergoes degradation. But the substrate of this tryptophan pyrrolase is the amino acid tryptophan. This tryptophan stabilizes this enzyme tryptophan pyrrolase, and so tryptophan can decrease degradation of tryptophan pyrrolase. Now, biological importance of this is that when you take a tryptophan rich diet, at that time more of the metabolism of tryptophan is required. So by tryptophan make it sure by stabilizing its metabolic enzyme, that is tryptophan pyruvate. So this is also a very well regulated metabolic regulation. So with this, I complete the regulation of enzyme activity in a living system. I hope that all the concepts are clear. If you have any query or confusion, please write it down in the comment section below. Thank you.